A quick recap of what you did yesterday before I introduce today's work. I'm going to go pretty quickly. If you need more explanation or you haven't understood something, come and talk to me today in the Google Meet. So you should have watched this video yesterday. The link was in your worksheet. If you've not, please do so. And yesterday we looked at asexual reproduction as being offspring you get from one parent. And we said that they were identical to the parents and that was called a clone, and that they were produced by mitosis because the parent organism is just growing its own cells to make a new organism. Therefore, the DNA is identical, except for some sort of environmental influences that might change the presentation. But effectively, they're identical to the parent. You went through the advantages and disadvantages. I've summarised in the table there what's in your book, so it's very efficient. You don't need a mate. You can produce lots and lots of offspring, so lots of organisms use it. Mostly plants and fungi use it, but there's a big problem with it being a monoculture, as in there's no genetic diversity, they're all the same. And we saw with the potatoes that if a disease comes through, because there's no genetic diversity, they all died. And the only way you get any sort of variation is through a mutation that might happen in the population. So hopefully you understood that yesterday and you summarised it well. And then you looked at the potato famine, this idea that I'm just going to click through these slides because you should have read it yesterday. So the idea that the Irish people had their little bit of land that they planted potatoes on, that they had planted this one sort of potato, and when the blight came through, this fungus that destroyed them, because they had this one cloned potato. I'll have a look at this on a slide that's coming up, but because they were genetically identical, when the fungus came through, they all got the disease, they all died, and millions of people starved. Had they planted a variety of potatoes, this wouldn't have been a problem, because probably only the lumpers would have died. These other ones might have survived and they would have been okay. So if you have a look down here at this tube, this is how potatoes grow. They grow off a central stem. The potato plant's up here, and you get all the potatoes genetically identical down here. And if you've seen old potatoes in wherever you keep your potatoes, they get those eyes on them that grow a new plant. Stick those in the ground to get a new potato, and that's pretty well how the Irish grew their potatoes from one. So you looked at fission, budding yesterday. You didn't look at fragmentation. We'll look at that today. You looked at spores and you looked at vegetative propagation, but you did not look at parthenogenesis. So we'll have a quick look at that today. Okay, so have a look at the little diagram here. This is basically binary fission. Copy the DNA, 2 cells. We learned about that last time in our last topic. So asexual reproduction, where every organelle is copied and the organism divides in 2. If we're talking about bacteria and prokaryotic cells, the only organelles that they don't have organelles. All they're copying is the DNA and the brain's not working this morning, makes protein, ribosomes. Okay, you can have fission in single-celled eukaryotic organisms, but mostly what we're talking about is binary fission in prokaryotic cells like bacteria. So there's a little animation there that you can watch of binary fission in a protozoan, and another animation of bacteria speeding up. I'll lead you to go into the slideshow and do that. I'm speaking really quickly now. We chopped our planarian up and we saw that the head started to grow the rest of the body and the tail started to grow the rest of the body. Those cells are coming from the bit of the planaria that was left, so therefore via mitosis, so therefore this is a form of asexual reproduction. Starfish can do the same thing. Starfish can lose a limb and grow a new one, or the limb can actually grow into a starfish. So this is called fragmentation. I'm going to ask you to add that to your document that I give you today. Budding you looked at yesterday, where the organism grows another little mini-me off the side. Mitosis, same DNA, asexual reproduction. Watch the video. I'm not going to show it to you now. It's a little bit creepy because there's no sound, but please watch it. All right, so spores, a couple of people came back to ask about this. Please, on your walks, see if you can find a fern and have a look at the bottom of it. If you see these little brown pockets on the bottom of it, these are sporangia that have spores inside them. So these can be sexually produced but are usually asexually reproduced, which means made by mitosis. And when they land somewhere, so they're very light, they'll float in the wind or on water, whatever, they land in a new place and they can grow into a new plant because they're produced by cell division in the parent mitosis cell division. They're identical to the original parent organism. All right, so thick resistant wall, so they're pretty hardy things and they're often produced in these little structures called sporangia. So check out the bottoms of the fern to see if you can see these little brown things. And here's a video of a fungi, a mushroom. The gills underneath are where the spores are produced and stored, and this cloud are the spores coming out of the mushroom. So please watch that little video today if you've not already done so. Okay, you can read that. I'm just going to come to this one. So parthenogenesis is this idea. Here's the queen bee. She's got two ways of reproducing. She can do the normal, her egg is fertilized by a sperm. It's diploid because it's got her set of chromosomes from the egg and the set of chromosomes from the sperm. Full set of chromosomes here in a diploid offspring that then grows up to be a worker bee or a queen bee. Or unfertilized eggs, which are haploid, have half a set of chromosomes, just the queens, can also develop into male organisms that are infertile because they've only got the one set of chromosomes. Alrighty, so the parthenogenic baby will always be a clone because it is identical, well, not really identical to the queen because it's got half a set of the chromosomes, but this can happen in these sorts of organisms. Sorry, I'm going really quickly. So parthenogenesis, where an unfertilized egg can develop into a viable offspring. So obviously the half a set of chromosomes that are there allows it to develop into an offspring that is viable, can survive. You looked at vegetative propagation yesterday. I'm not going to read all of this to you. There's some nice images on the next slide. So grafting's used sometimes for fun like this, where you graft, what have we got there? We've got limes and we've got lemons and we've got kumquats all on the one plant, but sometimes also used if we've got a really sturdy stock to graft less strong stock onto. We've got this idea like strawberries where they send a runner across the ground and then put roots down, grow another little strawberry plant. The runner can break here and we've got two new plants that are identical. We've got the bulbs growing off the side here, so you plant one daffodil one year and you get 4 daffodils the next year. And again, this grass reproducing by sending this long runner and growing new plants up off that runner. This is called vegetative propagation. Okay, so this is the new work for today. So you're going to read the textbook, you're going to listen to me, and hopefully you're going to understand. So here's 2 cows in a cow show. This is the winner, this one came second. This is a clone of this one. So at some point, the cells from this cow, who's called Apple Red ET, her cells were taken and grown in a laboratory in some way, implanted into a surrogate mother, and developed into Apple 3 Red ETN. Fantastic name. They are genetically identical. They've got exactly the same chromosomes inside their cells. There could be some slight differences depending on environment, how well they were fed. Maybe in the uterus there might have been a few changes, but they are clones of each other. So we're going to have a look at how animal cloning can happen, and here's the first way, embryo splitting. So this duplicates what happens when identical twins form. So here we've got fraternal twins, and here we've got identical twins. So what happens with identical twins? We've got the egg fertilized by the sperm, It's inside the zona pellucida. It separates into two cells, and then something happens to split that into two individuals. These are identical because they have come from this one egg and this one sperm, as opposed to fraternal twins who come from two separate eggs, two separate sperm, two separate lots of DNA. So in animal cloning, where they use this procedure, they use like an IVF process to fertilize an egg in a petri dish. They remove the protective coating around the cells that are produced. They separate the cells. So now you've got two separate lots of organisms growing that are identical to each other and can be implanted into different animals to make two identical organisms. So here we've got a bunch of cells up here, early embryo. In the laboratory, they separate some of these identical cells out of the blastocyst, separate them into the individual cells, let it grow to be a little bit of an embryo, and then implant them into surrogate mothers. So it doesn't matter what the mother looks like, the offspring are going to be identical because they've come from this original embryo. So this is called embryo splitting. You start with one embryo, you split the cells into separate embryos, implant them into surrogates, and you get clones. The other way of doing it is called nuclear transfer, and there's a little animation for you to play with which will help you with this. So the nucleus is removed from an unfertilized egg cell. The DNA is replaced with the DNA from an adult normal body cell, a somatic cell, from either a male or a female donor. Electric shock fuses the nucleus into an empty egg. The egg is implanted into the mother or surrogate where it will develop. So here we've got the donor cell is taken from this sheep's udder. So here's the donor nucleus. That donor nucleus is then put into an empty egg. So we have to suck the nucleus out of this egg. So here's the egg that came from this sheep. So we want the DNA from this sheep. We can take it from anywhere. We take an egg from this sheep, take the nucleus out of it, and then we put the nucleus from this sheep into that egg. The nucleus, because it's from a somatic cell, from a body cell, we don't need fertilization. It's got a full set of chromosomes. It's inside this egg now. It grows to be an embryo. We implant it into a surrogate sheep, and we get a lamb that is identical to this one. I know this sounds a bit confusing. So you're going to have a go at this. You're going to click on this link and follow the instructions to click and clone, and it will work you through how to do that. All right, and that's the end of my presentation.